Welcome to G Fun Facts Online. Today we are dissecting one of the most profound shifts in the history of biological science. For as long as humanity has feared death, we have looked to the blood of the young as a potential salvation. It is a concept that stains our mythology, from the vampire legends of Eastern Europe to the apocryphal tales of Countess Elizabeth Bathory bathing in the blood of virgins to preserve her beauty. For centuries, blood was seen as the carrier of the soul, the vital humor that dictated health, temperament, and lifespan. But where mythology saw magic, science eventually saw mechanism. The transition from folklore to the laboratory has been long, gruesome, and fascinating. But in the last decade, it has accelerated at a pace that has stunned the scientific community. We have moved from the crude, macabre experiments of stitching animals together to the precise, molecular identification of the factors that actually drive rejuvenation. We have found the ghost in the machine. It is not magic, it is not a soul, it is a protein. This is the story of platelet factor 4, or PF4, a humble molecule known for decades only as a cog in the blood clotting machinery, which has now been revealed as a master key for reversing biological aging. It is a story that unites three separate rivers of research: young blood transfusions, the longevity hormone Klotho, and the benefits of exercise, into a single powerful delta of understanding. We are no longer just slowing the clock, we are learning how to turn it back. To understand the magnitude of the PF4 discovery, we must first understand the black box that preceded it. For nearly a century, scientists knew that young blood could rejuvenate the old, but they had no idea why. The scientific investigation into blood-borne rejuvenation began in earnest in 1864 with the French physiologist Paul Bert. Bert pioneered a surgical technique called parabiosis, from the Greek para, meaning next to, and bios, meaning life. He carefully sutured the flanks of two albino rats together, connecting their capillaries so they shared a single circulatory system. It was a crude attempt to create artificial Siamese twins. Bert was studying graft compatibility, not aging. It wasn't until the 1950s that Clive McKay, a biochemist at Cornell University, applied this technique to gerontology. McKay was already famous for discovering that caloric restriction could extend lifespan, but he wanted to know if the factors of youth were transmissible. In a series of experiments that would become legendary and controversial, McKay performed heterochronic parabiosis, stitching an old graying rat to a young, vibrant one. The results were visually shocking. The old rats, receiving a constant infusion of young blood, began to change. Their fur became glossy. Their cartilage, usually brittle and yellowed with age, looked younger. They lived longer than their non-joined peers. Conversely, the young partners suffered. They aged prematurely, their growth stunted by the toxic influence of the old blood. McKay's work languished for decades, dismissed as too macabre or difficult to replicate. But in the early 2000s, a team at Stanford University, led by Irina and Michael Conboy, Thomas Rando and Irving Weissman, resurrected the technique with modern molecular tools. Their 2005 paper in Nature was a bombshell. They proved that the rejuvenation wasn't just cosmetic. Old muscle stem cells, which typically go dormant and refuse to heal injuries, suddenly woke up and repaired muscle tissue as if they were young again. The liver, too, regained its regenerative capacity. The conclusion was inescapable. Aging is not just a breakdown of the hardware, the cells. It is a failure of the software, the chemical signals in the blood. If you change the signals, you can reboot the hardware. This sparked a gold rush in Silicon Valley and academia. Startups began looking for the specific proteins in plasma that were responsible. Was it GDF11? Was it TIMP2? Was it something else? The field fractured into different camps, each championing different molecules. Some argued it was about adding young factors. Others argued it was about diluting old, toxic factors. The answer, as it turned out, would come from an unexpected convergence of three completely different fields. By 2023, the anti-aging field had three miracle interventions that were known to improve brain health and cognition in mice, but they seemed unrelated. First, there was parabiosis and young blood, as we just discussed, which boosted memory and neurogenesis. Second, there was Klotho injection. Klotho is a longevity hormone named after the Greek fate who spins the thread of life. Overexpressing it extends lifespan, lacking it causes accelerated aging. Injections of Klotho improved brain function, but there was a mystery. Klotho is a large molecule that cannot cross the blood-brain barrier. How was it affecting the brain if it couldn't get in? Third, there was exercise. It is the oldest and most effective anti-aging drug we have. Running increases new neuron growth in the hippocampus. But again, the question was how? What is the molecular signal that tells the brain the legs are moving? In August 2023, three independent papers published simultaneously in Nature, Nature Aging, and Nature Communications provided the answer. Saul Vieda's team at UCSF was looking for the factor in young blood. Dina Dubel's team at UCSF was looking for the messenger of Klotho. Tara Walker's team at the University of Queensland was looking for the exerkine released by exercise. They all found the same thing. When Vieira's team analyzed young plasma, they found high levels of a chemokine called Platelet Factor 4, or PF4. When Dubel's team injected Klotho, they found it stimulated platelets to release PF4. When Walker's team forced mice to run, they found their platelets activated and dumped PF4 into the bloodstream. It was a triple crown of biology. PF4 was the common denominator, the molecular funnel through which these disparate interventions channeled their rejuvenating effects. To understand why this discovery is so surprising, you have to look at what PF4 was thought to be. CXCL4, or platelet factor 4, is a small cytokine belonging to the CXC chemokine family. For decades, hematologists knew it simply as a promoter of blood coagulation. When you cut your finger, platelets rush to the site, activate, and release granules containing PF4. Its job is to neutralize heparin-like molecules on the endothelial surface, promoting clotting and calling immune cells to the wound to fight potential infection. It was viewed as a first responder, a grunt worker of the immune system. The idea that this clotting protein could travel to the brain, rewire neurons, and reverse the cognitive fog of aging seemed ludicrous. PF4 is now classified as an exerkine, a signaling molecule released in response to exercise. It joins the ranks of other famous molecules like irisin, released by muscles, and BDNF, brain-derived neurotropic factor. However, PF4 is unique because it comes from platelets. We typically think of platelets as lifeless sacks of glue, but they are actually circulating reservoirs of potent biological signals. They are the first responders of the body, and it turns out they are also the maintenance crew for the brain. In the elderly, platelet function changes. They become hyperactive in a bad way, leading to clot risks, but hypoactive in releasing beneficial factors like PF4. The levels of PF4 drop significantly with age. The 2023 studies showed that restoring these levels to a youthful range was sufficient to mimic the benefits of exercise and young blood. So how does a blood-clotting protein fix a forgetting brain? The research has unveiled a two-pronged mechanism, one systemic, involving the immune system, and one central, involving the brain directly. Let's look at the systemic reset first. Aging is accompanied by a chronic low-grade inflammation known as inflammaging. The immune system, which should be a precise sniper, becomes a clumsy shotgun, firing pro-inflammatory cytokines everywhere, damaging healthy tissue. Furthermore, the hematopoietic stem cells, or HSCs, in our bone marrow, which are the mothers of all blood cells, change their behavior. As we age, they stop making as many adaptive immune cells, like T cells and B cells, that fight specific threats, and start making too many myeloid cells, like macrophages and neutrophils, that drive inflammation. This is called myeloid bias. PF4 acts as an immuno-restorer. Recent studies in late 2024 and 2025 have shown that PF4 specifically targets aging HSCs in the bone marrow. It binds to them and tells them to stop overproducing myeloid cells. It restores the delicate balance between lymphoid and myeloid lineages. By reducing the flood of pro-inflammatory myeloid cells, PF4 effectively calms the systemic environment. It reduces the circulating factors that tell the brain it is old and inflamed. Then there is the brain connection, the hippocampal reboot. The brain is protected by the blood-brain barrier, a fortress that keeps most blood proteins out. However, the immune system and the brain are in constant dialogue. PF4 appears to work through the CXCR3 receptor. In the aged brain, microglia, the brain's immune cells, are often in an angry, inflammatory state. They eat healthy synapses and release toxins. PF4 signaling calms these microglia, switching them from a destroy mode to a nurture mode. The study showed that PF4 directly improves long-term potentiation, or LTP, in the hippocampus. LTP is the cellular basis of memory, the strengthening of connections between neurons. Perhaps most importantly, PF4 restores the ability of the aged hippocampus to grow new neurons. In behavioral tests, old mice treated with PF4 performed just as well as young mice in water mazes and fear conditioning tasks. They didn't just look younger, they thought younger. The role of Klotho in this narrative adds a layer of elegance to the biology. Klotho is a protein that is naturally produced in the kidneys and brain, but its levels plummet as we age. Genetic engineering that increases Klotho creates supermice that live 30% longer. Removing it creates mice that die of old age in weeks. For years, Dina Dubal at UCSF had shown that injecting a fragment of the Klotho protein improved brain function. But the fragment was too big to cross the blood-brain barrier. It was an action at a distance mystery. The 2023 breakthrough solved it. Klotho binds to platelets and commands them to release PF4. PF4 is the runner that carries the message. This implies a hierarchy of rejuvenation. First, the trigger, exercise or Klotho. Second, the mediator, platelets activate. Third, the messenger, PF4, is released into circulation. And fourth, the effect. The immune system calms and the brain rewires. This discovery is crucial for drug development. Klotho is a large, difficult-to-manufacture protein. PF4 is smaller and more stable. But even better, we might be able to develop Klotho mimetics, small molecules that simply tickle platelets to release their endogenous stores of PF4, bypassing the need to inject proteins altogether. If PF4 is so miraculous, why aren't we injecting it into humans yet? Here lies the safety paradox that separates mouse models from human medicine. In the world of hematology, PF4 has a dark side. To a doctor, anti-PF4 is a terrifying phrase. It is associated with heparin-induced thrombocytopenia, or HIT, a deadly reaction to the blood thinner heparin. In HIT, the immune system mistakenly attacks the complex formed by heparin and PF4. This activates platelets wildly, causing massive clotting throughout the body, while simultaneously dropping the platelet count. It is a clotting storm that can lead to limb amputation or death. Furthermore, during the COVID-19 pandemic, a rare side effect of adenovirus vector vaccines was VITT, vaccine-induced immune thrombotic thrombocytopenia. This was essentially autoimmune HIT, driven by antibodies attacking PF4. The challenge is clear. We want the regenerative effects of PF4, the immune calming and neurogenesis, without the thrombotic effects, the clotting. The therapeutic dose for rejuvenation might be different from the levels that trigger clotting. Biotech companies are currently racing to engineer super PF4 or PF4 fragments. They are looking for the specific part of the molecule that binds to the CXCR3 receptor on immune cells and neurons, while removing the part that binds to heparin or triggers the coagulation cascade. We are also looking at the route of administration. Systemic injection carries the highest risk. Could it be delivered subcutaneously, or perhaps via nanoparticles that target the bone marrow specifically? While we wait for a safe drug, PF4 is already proving useful as a diagnostic. Recent studies have shown that reduced serum PF4 levels correlate strongly with cognitive decline in humans. In Alzheimer's patients, PF4 levels drop in sync with the rise of amyloid beta and tau proteins. This suggests that PF4 could be a cheap, blood-based biomarker. Instead of an expensive PET scan or a painful spinal tap, a simple blood test measuring PF4 could tell a doctor that a patient's immune protective shield is failing and cognitive decline is imminent. The discovery of PF4 marks a paradigm shift in how we view aging. For decades, we treated aging as wear and tear, like car tires going bald. We thought we had to replace the parts with stem cell transplants or organ printing. PF4 suggests that aging is more like a software glitch. The car is fine, but the computer is sending the wrong signals, putting the engine into limp mode. The immune system is stuck in a loop of inflammation. The stem cells are stuck, making the wrong lineage. By introducing a restorer molecule like PF4, we act as system administrators. We clear the cache. We reset the parameters. The roadmap for the next 5 years is exciting. Phase 1 clinical trials will likely start with PF4 mimics or small molecule agonists of the CXCR3 receptor. The initial target won't be aging, which the FDA doesn't recognize as a disease, but specific conditions like mild cognitive impairment or frailty. We will likely see combination therapies. Just as HIV is treated with a cocktail, aging will likely be treated with a symphony of exerkines. Imagine a therapy that combines PF4 for an immune reset, irisin for a metabolic reset, and GDF11 for a vascular reset. This research also validates the critical importance of exercise in the elderly. It proves that the runner's high is not just endorphins. It is a flood of regenerative platelet factors protecting the brain. It provides a molecular mandate for keeping the elderly active. We have come a long way from Paul Bert's rats, stitched together in a gaslit French laboratory. We have moved beyond the myths of vampires and the macabre search for young blood. In PF4, we have found a molecule that bridges the body and the brain, the blood and the mind. It teaches us that the seeds of our rejuvenation are already inside us, locked away in our platelets, waiting for the signal, a run, a hormone, or perhaps soon, a medicine, to be released. The Immuno-Restorer is not just a hope, it is a mechanism, and mechanisms can be mastered. Thank you for listening to G Fun Facts Online.